So the team approach can be interpreted as teams of clinicians around the world that specialize in seeing patients, working together, putting them on trials. But it doesn't intend to dismiss the importance of translational scientists and basic scientists. Because if we're going to take the approach that we're going to start from scratch and look for new drugs and explore the merits of combinations, I wholeheartedly believe that needs to start in the laboratory. We need to be able to vet these combinations in reasonable surrogate models of the disease and then identify those combinations that have the greatest potential and then translate those to the clinic. When we move to the clinic, we really need to also focus on understanding the pharmacology of these drugs and how we think they're working and the context within which they are working. So are they working in this mutation or that mutation, this immunophenotype, that immunophenotype? So that requires us to go from the lab to the clinic, but just as important to take that information from those trials and do those correlative studies that take us back to the lab where we can add additional color on the exact molecular subtype of lymphoma these patients have and try to identify a particular biological context where these regimens might work better or not as well. It is incredibly rewarding to be able to give and develop a drug in the laboratory, study it in the laboratory, try to garnish as much insight into its mechanism as you can, and then to try to study those effects in vivo models. And there's a few things for me as exciting as giving a brand new drug to a patient that's never seen that drug before, a human being, and trying to really focus on how's the best way to give those drugs, making sure, of course, it's always safe first, then trying to optimize pharmacokinetics, pharmacodynamics, other pharmacologic parameters to make sure we get that part right before we move into the phase 2. So the translational piece, how do we move to the clinic? The phase 1 piece, have we optimized the pharmacologic properties of the drug? The phase 2 piece, have we identified the patient population within which we believe the drug is likely to have the greatest clinical benefit? And then moving into the regulatory piece. How do you think about the approval of a drug in T-cell lymphoma or mantle cell lymphoma or cutaneous T-cell lymphoma in the context of the current landscape of drugs that are out there is interesting and challenging, and it's always evolving. As we've now been able to get many drugs approved in mantle cell lymphoma, so since bortezomib, we now have lenalidomide and BTK inhibitors in mantle cell lymphoma, and we've changed the natural history of that disease with new drugs. And in PTCL, I'm certain we're on the verge of doing the same thing with a whole host of new drugs. So that dynamic changes as we approve drugs in the space, and what a regulatory study looks like in 2018 or 2019 may not be what it might have looked like in 2010 or 2013 because it's always changing. And so trying to have some foresight about what that regulatory study looks like and trying to think about the landscape and defining that patient population as precisely as you can is critical to the regulatory success.